Hello, and welcome to Intro to Water Applications with Hyperspectral Remote Sensing. In this episode of our Hyperspectral Academy series, we'll explore the critical role of hyperspectral imaging in understanding and managing our water resources. From assessing water quality to mapping benthic habitats, we'll uncover how this technology is pivotal in aquatic studies. Aquatic ecosystems, encompassing oceans, lakes, rivers, and wetlands, play a crucial role in supporting biodiversity and regulating global climate. They are instrumental in regulating the Earth's climate, including carbon sequestration and oxygen production. Aquatic ecosystems also play a pivotal role in nutrient cycling and water filtration, helping to break down organic wastes and detoxify harmful pollutants before they reach larger water bodies. Wetlands, often referred to as the kidneys of the landscape, are particularly crucial in this regard. However, despite their importance, aquatic ecosystems are among the most threatened environments due to pollution, habitat destruction, climate change, and overfishing. Effective management and restoration efforts are crucial, and hyperspectral remote sensing provides a powerful tool to monitor and assess the health of these ecosystems continuously and accurately. Hyperspectral imaging serves a broad array of applications in aquatic ecosystems. On one end of the spectrum, you have applications more focused to water quality, such as detecting and analyzing pollutants, to tracking algal blooms, and monitoring sediment transport. Hyperspectral data also improves our ability to classify underwater habitats and assess the health of coral reefs and seagrass beds. And not only that, it can better enable estimates of fish stock populations and assess habitat suitability, as well as better evaluate the effects of human activities on coastal and inland water ecosystems. Throughout the rest of the video, we will delve into the science behind some of these applications, demonstrating the technology's versatility and critical importance. Now, with hyperspectral remote sensing of aquatic environments, understanding how light interacts with water is crucial. Water bodies are complex optical media, where several processes affect the propagation of light, thereby influencing the spectral signals detected by remote sensing instruments. These processes include absorption, scattering, and fluorescence, each playing a critical role in defining the water's spectral signature. Let's look into this a little bit deeper. The water itself is a strong absorber of certain wavelengths of light, particularly in the red to infrared and ultraviolet regions, which is why when you have very clear water, it appears blue. However, when you get into more coastal and inland environments, the presence of other substances, such as phytoplankton or sediment, significantly alters these absorption characteristics. For instance, the pigment chlorophyll A, found in almost all living plants, is also ubiquitous among tiny microscopic plant-like organisms called phytoplankton. And similar to typical vegetation, they absorb light predominantly in the blue and red wavelengths, facilitating photosynthesis by absorbing CO2 and producing oxygen, just like plants. And its presence can indicate productivity levels in the water. And actually, it's estimated that phytoplankton contribute anywhere from 50 to 85% of the oxygen in Earth's atmosphere. It's hard to emphasize just how important these little critters are to the functioning of our planet. Now, in open oceanic environments, phytoplankton is the main contributor to changes in the water color. As phytoplankton biomass increases, we tend to get greener water. Scientists typically call these waters case 1 waters. And as we get closer to land, we start to see other competing spectral signals from other substances. These are called case 2 waters and encompass waters where other optically active substances can affect the light field. One of these substances being color dissolved organic matter, or CDOM, also known as gelbstoff or yellow substance. It is comprised of complex organic molecules that result from the decomposition of biological materials like leaves, roots, and phytoplankton. These materials leach into aquatic environments from soils, marshes, and coastal vegetation. CDOM absorbs light strongly in the ultraviolet and visible blue spectra, which significantly reduces the penetration of these light wavelengths through the water. In fact, in instances of very high CDOM loads, the water can sometimes almost look black. This absorption can influence various ecological processes in aquatic systems by affecting photosynthesis as less light reaches the water's deeper layers. CDOM also plays a crucial role in the aquatic carbon cycle by binding with heavy metals and pollutants, affecting their transport and bioavailability in water bodies. Another common component of case 2 waters are suspended sediments. These refer to the particles such as clay, silt, and organic matter that are carried by water currents and do not readily settle. Optically, these sediments are highly significant because they strongly scatter and absorb light, which can greatly affect the clarity and color of the water. Similar to CDOM, they tend to absorb more strongly in the UV and blue, but depending on the content, different sediment types can have distinct spectral responses. Hyperspectral imaging can distinguish these differences, helping in the classification and quantification of different sediment types. For instance, clay might impart a yellow or red tint, while organic-rich sediments would give a brown or green hue, depending on their composition. They are also important indicators of water quality and ecosystem health, as they can carry nutrients, pollutants, and pathogens, influencing the biological and chemical processes in aquatic environments. Hyperspectral imaging can then help trace the distribution of these pollutants by monitoring sediment movements, as well as understand how sediments move and inform erosion control measures, sediment management in reservoirs, and habitat restoration efforts, especially in riverine and coastal environments. Now that we have a better understanding of some of the optical theory behind how light interacts with water, let's look into some of the practical applications of how HSI can be used in water quality monitoring. Algal blooms, particularly those involving cyanobacteria or blue-green algae, can have significant negative impacts on water quality, aquatic life, and human health. These blooms are often driven by an excess of nutrients like phosphorus and nitrogen, which can result from agricultural runoff, wastewater discharges, and other anthropogenic sources. Here we are showing a Pixxel satellite image of Provo Basin at Utah Lake, an area which is frequented by algal blooms, some being potentially toxic. We can visually see areas of intense green, signifying elevated levels of phytoplankton. But let's see how we can leverage HSI to understand the amount of phytoplankton present, and even be able to differentiate if it is potentially toxic. In highly productive waters, a certain region of the electromagnetic spectrum is particularly useful, the red edge region, which is the boundary between the red and near-infrared parts of the spectrum, approximately 680 to 730 nanometers, and is particularly sensitive to changes in chlorophyll content. Areas with high chlorophyll A concentrations show a pronounced shift in the red edge peak position towards longer wavelengths. Let's look into why. As previously mentioned, chlorophyll A absorbs strongly in the blue and red, but there is another fine-scale feature hyperspectral can exploit that we haven't talked about yet, and that is the sun-induced chlorophyll A fluorescence. Chlorophyll A fluorescence is a natural emission of light by chlorophyll when algae absorbs sunlight. And this phenomenon occurs because some of the absorbed light energy is re-emitted at specific wavelengths, providing valuable insights into the photosynthetic efficiency and photophysiology of the organism. Phytoplankton fluoresce at a very specific region centered at about 685 nanometers. And this becomes very evident as phytoplankton concentration starts to increase at very early stages of some blooms. However, as concentrations become very large, the absorption of phytoplankton at 675 nanometers from chlorophyll A and the strong backscattering signal in the near-infrared, combined with the strong absorption of water in the near-infrared, creates a significant scattering-induced peak around 710 nanometers. Thus, with high spectral and temporal HSI, such as Pixxel sensors, we can accurately track and quantify these blooms from initiation stages where fluorescence may dominate the red edge signal to intense blooms as we see here, where the strong scattering peak near 710 nanometers starts to dominate. Let's see what else we can uncover from this image that may help local environmental managers. I mentioned previously that cyanobacteria is known to frequent this lake. Commonly known as blue-green algae, cyanobacteria are photosynthetic microorganisms found in freshwater and marine environments, and they can proliferate rapidly to form harmful algal blooms, or HABs, that produce toxins detrimental to aquatic and human health. Thus, it is extremely important to be able to detect, monitor, and predict when these blooms are going to happen. Luckily, cyanobacteria have some distinct spectral features that we can exploit to be able to identify them. Unlike other phytoplankton, they contain the pigment phycocyanin, which contains a strong absorption peak centered at about 620 nanometers. Thus, with an HSI system such as ours, we can exploit this feature and quantify the amount of absorption happening in this region, which would signify that this bloom is indeed cyanobacteria, and specific alerts and health advisories can be broadcast to the local community. Aside from direct health impacts, algal blooms can have significant and complex impacts on carbon cycling and carbon fluxes in aquatic ecosystems, influencing both carbon storage and release. Algae, including cyanobacteria that form blooms, are primary producers that use photosynthesis to convert CO2 into organic carbon. This process draws down carbon dioxide from the atmosphere or water and contribute to short-term carbon storage. During bloom events, this uptake can be substantial due to the high biomass of algae. This carbon becomes part of the phytoplankton biomass, essentially storing carbon in their cells, which vastly increases the backscattering signal of these organisms. Using advanced HSI technology in conjunction with advanced modeling, we can separate this algal backscattering signal from other scattering sources, such as from sediments, and derive estimates of the amount of organic carbon present in the water, as you can see here. By understanding the carbon content and dynamics of phytoplankton populations, scientists can better model and predict carbon fluxes in aquatic environments, which are crucial for understanding global carbon cycles and assessing the impacts of climate change. For effective remote sensing, understanding these interactions is key. It allows researchers to design better algorithms and models to interpret hyperspectral data accurately. For example, algorithms can correct for the effects of scattering and absorption to better estimate the concentration of chlorophyll or suspended sediments. Hyperspectral imaging has been pivotal in several groundbreaking environmental studies and conservation efforts pertaining to aquatic ecosystems, beyond just assessing water quality. Benthic habitats, such as coral reefs, seagrass beds, and sandy or rocky substrates, play crucial roles in aquatic ecosystems. These habitats support diverse marine life and provide services such as nutrient cycling and carbon sequestration. Hyperspectral imaging offers detailed and precise tools for classifying these habitats and monitoring their health, allowing researchers and conservationists to make informed decisions about protection and restoration efforts. Each type of benthic substrate has a unique spectral signature. Hyperspectral sensors can capture these fine differences in reflectance and absorption characteristics at narrow spectral bands. By analyzing these signatures, scientists can map different habitats and assess their spatial distribution and fragmentation. Particularly, coral reefs are highly sensitive to changes in water temperature and quality. Coral health is intricately linked to a symbiotic relationship with microscopic algae, similar to those found in the water column. These algae reside within the coral tissues themselves, providing corals with essential nutrients via photosynthesis, which is critical for coral growth and reef building. They also provide the corals their distinct colors, and when corals get stressed, they will dispel these algae from their tissue, turning the coral more white. This is known as bleaching, and leads to weakened health and increased mortality risk. Hyperspectral imaging can detect early signs of stress, such as bleaching or disease, by identifying shifts in spectral signatures associated with coral pigmentation and health. This early detection is vital for initiating timely conservation actions. Seagrass beds are another benthic type that are vital ecosystems that stabilize the seafloor, improve water clarity, and provide habitat for diverse marine life. They also play a crucial role in carbon sequestration and nutrient cycling. Hyperspectral imaging is instrumental in monitoring seagrass health and distribution, as it captures detailed spectral data that can differentiate seagrass from other aquatic vegetation and detect stress signals before visible signs appear. This allows for timely management actions to protect and restore these critical habitats. By safeguarding our aquatic ecosystems, we protect not only an invaluable ecological treasure, but also a critical economic resource that millions of people worldwide depend on for their livelihood and survival. Hyperspectral imaging, rich in data and information, stands at the forefront of this endeavor, offering insights that are indispensable for sustainable management and conservation efforts. Thanks for tuning into this episode of Aquatic Use Cases for Hyperspectral Imaging, and I hope you enjoyed it.